Okay, so this is test review part 2, and we're going to look at this one, and then we're going to look at this one. Okay, so starting here, it says transcription. Well, we know that transcription is it's the first stage, so it's number 1, it's the first stage of protein synthesis. What happens in transcription? The DNA that you find in the nucleus opens up and is used as a template to produce a strand of messenger RNA. Where does it happen? Well, clearly, because you're dealing with new DNA, and the only place you find DNA is in the nucleus. That is where transcription happens. And what is produced? Messenger RNA. So, and a complementary strand, which means that whatever the sequence of bases were in the DNA, the complementary sequence of bases are produced in the messenger RNA. All right, coming over here to translation. What happens in translation? That messenger RNA is used to produce a protein. Okay. What is a protein? It is a lot of amino acids which are all joined together in a specific sequence. The sequence of the amino acids in the protein control the sequence of, or the shape of the protein, and since the shape of the protein determines its function, it is very, very important that we get the amino acids in the right order. Where does it happen? Well, if you look here, you can see there's kind of a faint outline of where it happens. It happens in ribosomes. Now, what is the final product? It's a protein, okay? And that's typically what we call a sequence of amino acids joined together. But likewise, until it's not really a protein until it's actually got modified by the Golgi body. So until it's a protein, it's all been, folded and organized into the exact right shape for the job it's got to do. Often it's referred to as a polypeptide. So what are the two stages of protein synthesis? First, we have transcription, where the DNA is used to make a strand of complementary messenger RNA. And then secondly, we have translation, where the messenger RNA is used to assemble amino acids, right, AA there, amino acids in a specific sequence to make a protein or a polypeptide. Okay, let's now look up here. Okay, and I want to go through this step by step so that there's a lot of big test questions involve this part here, and I want you to just remember what all the stages are. So what I've got here clearly is a sequence of nitrogen bases, and obviously it's DNA, and the reason it's obviously DNA is I have T's in it. So if there is a T, thymine, it has to be DNA. So what's step number 1? I've told you this plenty of times. Break it down always into 3s. So we'll go ahead and do that. So transcription would be taking this strand of DNA and using it to make a complementary strand of messenger RNA. So if there is a T there, there is going to be an A here. A G is going to become a C. A C will become a G. Over here, the G becomes a C, the C becomes a G, and the T becomes an A. Here, an A, because we're turning it into messenger RNA, becomes a uracil, a U. A G will become a C, and a C will become a G. Okay, that is going to be transcription. The next step is which amino acid is coded for. So I need to use, since we've said this plenty of times, I'm going to go back over here to my codon chart. I'm going to just bring that in side by side here. So if my messenger RNA is ACG, A is the first letter, C is the second letter, they meet in here, and the third letter is G, so it's going to be that one there, threonine, THR. Okay, then I have CGA. C, G, and A, that's arginine, ARG. And then I have UCG, first letter U, second letter C. It's in this box. G is going to be serine. So I always remember to break it into 3, because these 3 in DNA is a gene and 3 in messenger RNA is a codon. And as you can see, this is the codon chart. So we have to use the codons to make sure that we get the correct amino acids. All right, let's move along. Let's take care of these two. I'm going to leave this codon chart over here for a moment. So the next one says, what is a mutation? And we spent some time talking about, the kind of crazy stuff that we think mutations are, but they're really not all that, complicated, because a mutation is quite simply, if you change the sequence of bases, change the sequence of nucleotides, since the nucleotide is ATGC, right? If you change the ATGCs, what you're going to do is you're going to change the messenger RNA, and that has the potential to change the amino acid that's produced as a result of the mutation. So in its simplest form, what is a mutation? It is a change in the sequence of nucleotides, that's the ATGCs, and of course that will then, if you change them, you're going to end up with new or different amino acids. And if you change amino acids, you are going to change the protein. And maybe that protein won't fit and won't work, and so it can't work properly, like we talked about with lactose intolerance, or maybe it will give you a whole new characteristic which actually could in some respects make things better for you. Typically that isn't the case, but it can be. So a mutation is the change in the sequence of amino acids, or the change in the sequence of bases, which leads to new amino acids and so new proteins. Okay, however, silent mutations, that's what we need to look at this for, because a silent mutation, it's still a change, right, in the ATGCs, change in the bases. But for example, what if you had CUA? Okay, what does that code for? CUA codes for CUA, that codes for leucine. Okay, what if I change this, right, to let me get this right, UUA. Can we get it right? Yeah. What if I changed it to UUA? So my mutation, what's changed is this first letter. I've gone from a C to a U. Okay? But when I look at what U, and A is, guess what it codes for? It still codes for leucine. So even though I've changed one of the bases, I'm still going to get the same amino acid, because you'll look and you see leucine here has got 1, 2, 3, 4, 5, 6 different codons that would code for it. So sometimes You can get these changes in the bases. It's still a mutation, but it's silent because it doesn't have any effect on the final protein. Alright, let me move back to, let's look at this one right here. We have a picture of a mouse. I'm sorry, I've got my cable's all tied up. I've got a picture of a mouse, and I've got a picture of a plant, okay? And it says universal genetic code. I feel like my mouse needs to have little pink ears. Okay, and maybe it's one of those weird mice with red eyes and creepy ones. So, what does the universal genetic code mean? It means that all organisms have the same components to make up their genetic code. So this mouse, let's look at this DNA sequence here. It's a gene because there's 3 DNA. I know it's DNA because there's a T. So if that was messenger RNA, that's going to become A, G, and a U, isn't it? And in the plant, TCA, in messenger RNA, is also going to be A, G, and U. Guess what A, G, and U codes for in a mouse? It codes for serine again, right? So this codes for serine. I only have one codon chart. This codon chart, let me get to it. This codon chart here, there isn't a codon chart for mice and a codon chart for plants. The codon chart is a codon chart. So AGU in a plant still codes for serine. The universal genetic code says, you know, all living things have DNA, and the DNA is all made up of the same types of nucleotides, and those nucleotides are transcribed into the same amino acid strands, and that AGU is always going to code for serine no matter what organism it is. Where it's different is that if you, this is just like a little snapshot here. If you took all of a mouse's DNA, you would have ATGCs in totally different order than all the DNA ATGCs in the plant. So, but any given gene, three bases in DNA, is going to be equivalent in the codons and in the amino acids that they code for. That's what we mean when we say that we have the universal genetic code, and all of these code for the same amino acids. Are you kidding? No.